Can they hear me? Nice. Hello, everyone. I'm so excited for you all to be here today. Um, my name is, if you don't know me, is Shirsheen Jobert, and I'm a cell biologist and the event coordinator of our first ever Atlanta chapter of Taste of Science, and I'm going to be your host for tonight's event. First, I want to thank you all so much for joining us. It truly means a lot, especially because this is our first ever Taste of Science event and we're doing it virtually, and we just really want to kick off this new Atlanta chapter, and our team is really excited to have you all here. And our journey started when our chapter lead, Luke Shoemaker, sorry about that, had the vision to bring science directly to the greater Atlanta community, and the idea grew into a partnership with Taste of Science, where we now are hoping to bring talks throughout the Atlanta community and making science more accessible and engaging for adults. So first, our mission is clear, and it's to bridge the gap between the scientific discovery and everyday life, connecting researchers with our community full of curious minds and science enthusiasts alike. And in the future, we can't wait to bring these events to you in person, right in your local community, at a coffee house, a brewery, any space we can get. But for now, we're starting digitally, so we're so glad that you're here for this very first step of our journey. And please feel free to comment your name in the chat so we can see who's present. I can give a shout out. Oh, but I can't see the chat. Oh, cute. Oh, thanks, Frankie. All right. And so I'm so excited and honored to introduce you to our first speaker, or our very first speaker for Atlanta Taste of Science ever, Dr. Ahna Skop. And now Dr. Skop is a renowned geneticist, artist, and a professor at the University of Wisconsin-Madison, and she is widely recognized for her groundbreaking work on cell division and her creative approach to scientific research and communication. She leads innovative research which focuses on the molecular mechanisms of the midbody and developmental and diseases, and her contributions have earned her prestigious awards, including the Presidential Early Career Award for Scientists and Engineers and the UW-Madison Chancellor Inclusive Excellence Award. But beyond her research, Dr. Skop is a passionate advocate for diversity in STEM, which is really important in today's society, and a leader in integrating art as well as science to engage and inspire broader audiences. And throughout this talk, please feel free to drop your questions throughout. Our moderators will keep track of them, and then we will have a Q&A at the end of this talk. And to kick things off, we have a quick question for you. So please, in the chat, answer how many of you know that mitosis produces 2 new cells? So go ahead and share your answers, and then we will keep track of that as the talk goes on. So please give a virtual round of applause to Dr. Ahna Skop.
Yay! Thank you for having me here. I'm excited to tell you the story of work that has come out of my research in my laboratory by lots of amazing students. And are we going to share my slides? Yep. So I'm going to tell you a little story about one cell dividing into 3 things, because many of you probably know that one cell divides into 2 things. And I'm going to start out with talking about things that you probably know about cell division. For me, someone who came from a family of artists, I became very drawn to cell division because it was beautiful. But then I also wanted to understand how this process works. And many of you appreciate that cell division is 1 cell going into 2 cells is very important for development. So after sperm and egg meet, the magic happens and cells start to divide. And this is what allows us to become the adults that we are and the humans that we are. And many of you probably do appreciate that when this process goes wrong, you get tumor and birth defects. And this is why scientists actually study this process, because we want to help people who have tumor and birth defects. What you might not appreciate is that under periods of lots of inflammation, patients often have multinucleate glial cells, which are cells in the brain that help neurons out. And these cytokinesis defects, I think, is one aspect of this Alzheimer's disease. And so this process, we not only can understand cancer and birth defects, but also has to do with neurodegeneration. So I asked the question about one cell's dividing into two things because most people know that's true, and this is the image that you've all probably been seeing. But I'm going to focus on the thing called the midbody, which people may not appreciate. And this actually shows up in a stage called telophase. And telophase is right before the cells separate. And so inside, so if you look at a cell dividing, there's a little canal that connects those two cells right before those cells separate. So this is an image of a human cell. It doesn't look like those nice drawings where it's very spherical. These are cells dividing, and they often crawl away from each other. Inside of that, if you zoom in, this is where the midbody sits. And so this is an image taken, electron microscope image taken of the midbody by Michael Mullins. And this is a structure that forms at the end of mitosis that was, it actually caused, people didn't understand what it did, but it was very interesting. And so people wanted to study it at that point, but it had this very unique structure. This whole thing is called the midbody, and it has this little very black dark space called the midbody matrix. And the image up in the corner is showing that there is actually a cleared out area. It's often called the dark zone. But these midbodies are found not just in human cells, but go all the way back. So if you look at a phylogenetic tree, this is how all species are related throughout evolution. Midbodies are found all the way back to sponges and up to humans, and they all have this electron density. And so this is an image of a midbody in sponge, and this is a picture of a midbody in a mouse embryo. And one of the newest midbody labs, Agatha Shane, had categorized that a lot of people had noticed this throughout different species, but it hadn't really been appreciated actually what this midbody does. The very first person to actually identify the midbody was Walter Fleming in 1891. He was the first person to discover it. Scientists were also artists. They had to sketch what they observed under the microscope. This is a picture of his microscope, likely that he was using at that time. He's also quite famous for coining the term for mitosis, so we have Fleming to thank. And in 1891 is the very first paper on the midbody. And I wanted to show you what a picture, an image that he took from this crude microscope. And there was a very insightful, these drawings that he had, I had looked at when I was a student. And I kept looking at that. And oftentimes the answers are always there. You just have to understand what you're looking at. But what Fleming drew here is he drew the nuclei, but he also noticed cells when they were in this division process. He also can observe this a little, there was a refractive, the way the microscope detects a refractive image is he could see this midbody. So he could see the nuclei and the midbody lit up. So that was kind of the hint. And he thought this was really important. This is how cells divide. Human cells, but also plant cells may use this structure. And that was kind of what was known in 1891. Amazingly, about 80 years later, someone else picks this midbody up. So it was kind of forgotten for science, which I think is kind of an interesting story. I really like the history of science. I think it's important to understand that we work with each other over sometimes hundreds of years to kind of solve a problem and understand how this process works. This is the case for the midbody. And so fast forward about 80 years, Breck Byers takes the first electron microscope image of this and sees this electron density. And what I wanted to point out on this slide is that many of you probably have heard of telophase, but what Byers had found is that telophase is much more interesting than meets the eye, is that the cell will actually round up, flatten, round up, and then eventually the midbody will clip away and that the cells separate. And that was what was known in the late 1960s. Fast forward about 15 years, and not many people worked on the midbody. There was about 2 around in the 70s and 80s. Ryoko Kuriyama is another famous midbody luminaire, is that she actually isolated these midbodies, injected them into rabbits, and was able to identify the first protein marker that was only labels the midbody, and this is in this bottom panel here. This was a kinesin protein. It's called CHO1. It's also called mClip1. And now, more recently, it's been categorized another name called KIF23, which you may hear throughout this talk. And this is very distinctive and only labels the midbody. And so when people are studying and looking for markers, it allows them to study not only the structure, but how this process works, and enables science to advance by doing that. So she's very famous for identifying this protein, and it's the first midbody marker protein that now turns out to be a pan-cancer marker. So when I joined graduate school in 1994, so about 10 years later, I joined the lab of someone who was a microscopist, and I saw this structure called the midbody, and I turned to my mentor and I said, "What is this thing?" And he turned to me and he said, "This is a garbage can." And so I actually was amazed by this. And there was a lore in the scientific community that people thought this was very not important. And Ryoko, who we have become close friends over this, and colleagues, is that she could never get any grants funded for this research in the 70s, and was told not to work on it because people thought a bunch of proteins go there and it's really not important. So it's a garbage can. I became quite fascinated with this story, and I realized there must be something more in this structure that meets the eye, and it can't possibly be a garbage can. So I decided to focus my entire life and career on this garbage can structure. And so I went off to do my postdoc at UC Berkeley. And around that time, there was an advance in science called proteomics. This is basically sequencing proteins. And so what I did with the help of Ryoko and other people, Michael Mullins, that I've cited here in these papers, is isolate these midbodies. And then we use a technique called mass spec, which allows we to sequence all the proteins within the midbody. And we use computational methods called bioinformatics that allows us to identify all the proteins that are in this structure. And then what I did is to take those proteins and find similar proteins in an organism called C. elegans, and then I knocked them down, and I was able to make videos on a microscope. And that's what you're seeing here. What I was able to do is identify all of those proteins and then go look on the microscope if they have cell division defect. And so what this experiment taught us is that it's actually not it's not a garbage can, is that we identified over 100 new proteins involved in cell division, and a total of a number of about 577 came out of that. And so that's how I started my lab here at the University of Wisconsin-Madison. So after my paper was published and on to the present day, what became very clear to everyone is that there were lots of proteins involved in disease. And I've color-coded this for you for ease, is the blue ones are involved in cancer, and the green ones are involved in neurodegeneration or brain cancers or gliomas. And you may know about one of them, microcephaly, which Zika actually causes microcephaly. And so it's unclear how all this works. But there was a protein called that is in the midbody that when you mutate it cause a very similar phenotype. And so that led us to believe that the midbody is very, very important for our understanding of human disease and to help. And so that allows us to make more important arguments for our funding and to be able to study this even further, which is what I've been doing in my lab for the last 20 years. And so my lab takes several approaches to understanding things in the midbody. We take this proteomics approach, which is sequencing all the proteins. And then we also look at the RNA in there. And so that's called transcriptomics. And so it's just basically sequencing either protein or RNA. And so we initially looked at the proteomics work here and all the proteins in the midbody. And what we found was a big surprise. So often in papers, there's lots of materials and they end up in something called a supplement. And in this supplement was a list of proteins, and there were 99 RNA binding proteins. So these are proteins that bind to RNA and maybe perhaps help their function. But they were categorized quite interestingly. That was unusual for the cell division process, is that they were involved in neurodegenerative disease. And that at the time, and also when I was definitely younger, I didn't know the connection between cell division and anything to do in a neuron. So what could be going on, right? So that was really interesting to me. And we wanted to figure out, maybe there was a hint in the knowledge of knowing that there were these RNA binding proteins and they have some function in the brain. And so I decided to study one of these, and this is called ataxin-2. Ataxin-2, when you mutate it in humans, causes spinal muscular ataxia type 2. Long story short, this disease is like a Parkinson's disease. It's ataxia, it's a shaking disease. And so that was interesting to me, is that something involved in cell division actually would cause this neurological disorder. This protein actually binds to RNA, and it happens to bind to RNA. So when you are under stress, it forms these little granules in the cell, and it stops them from being translated into protein. And so oftentimes you need to store things for later, and that's what this protein kind of does. But how does it function in mitosis? And so I went to look at this in an embryo of this nematode worm that I studied. And so the top panel shows you that in about 15 minutes, this embryo is dividing into 2 cells, and we can see this quite clearly. So the green is marking the plasma membrane, and we can look at the whole cell cycle quite easily, and the red is marking the DNA inside, so we know the state of this. And to our amazement is that when you knock down ataxin-2, you get 2 things happening that were quite unusual. One, you got definitely a cell division defect. So you end up in this last stage where you have multiple nuclei. That's a clear cell division defect. So instead of 2 cells, you forming 2 cells, you actually get a bag, one big cell with a bag of nuclei in it. So that's very indicative of a cell division defect or a cancer or what have you. But the other interesting phenotype is that there was a delay. So if you look at the timing of when the division happens, this happens in a normal embryo quite early, but it's delayed here. And this is happening at a different point in the cell cycle. So that was unusual because no other mid-body protein ever had this kind of phenotype. So we scratched our heads and kind of thought about what possibly is this telling us? So we put, scientists often put things, we get little snippets of information, and I always consider these little puzzle pieces. So we knew we had 99 RNA binding proteins that are in this midbody. The other thing is we knew there was these neurodegenerative disease targets in here, which was another piece of the puzzle. The last piece of the puzzle was this ataxin-2 story, which functioned in cytokinesis. And we had proposed that perhaps if you have RNAs there, they would be translated. And translation is involved in making proteins, which is how our body functions. And so perhaps this was a crazy idea to think this, but we were kind of thinking that this is perhaps what's happening because there was knowledge of this happening in the brain. And so we sought out to figure out and piece these puzzles together and to do more experiments in the lab to try to figure this out. And so my lab decided that maybe there's RNAs in the midbody. If we have all these binding proteins for RNAs, let's look at that. So we went to do transcriptomics, which is sequencing the RNAs. But before I do that, I wanted to go back to this one cell, title of my talk, dividing into 3 things. So I showed you this. I think most of you understand that one cell divides into 2 things. This is not entirely true. This is actually what happens is there's 2 cells are formed, and then you get a midbody remnant, which we call affectionately the MBR. And so the MB becomes this MBR, and instead of cleaving right down the middle, you get 2 cuts on either side, and you get this very large vesicle generated. And I myself didn't even know this actually existed until much later in my career. And a lot of people don't actually appreciate it quite a bit, but it will become very important as you will see. If you look at 2 cells dividing, and so what you're looking at here is a video. There's stills to the side here is 2 cells are dividing. These are the daughter cells that you probably learned about in school. The 2 red cells are labeled, and then the midbody is being labeled and jettisoned out. And you can see that other cells are taking this up. And when we first saw this video, this was back in 2014, this was quite exciting, is why would the cell do this? And what's happening when the adjacent cell is taking it up? Perhaps this is a clue for our understanding of cancer. The other little interesting bit that happened more recently from the Simpson lab at Monash University in Australia is what they noticed is that if you use an antibody that allows us to see the midbodies, is that in normal colon tissue, you'll see that midbodies are shed, which are labeled in red here. But if you look at colorectal cancer tissue, what you'll see is tons of midbody remnants are shed. And so this was quite an amazing thing to see, is that perhaps when you have cancer, you're shedding lots of midbodies, and that's indicative of disease. And these midbodies perhaps are moving around the body and causing cancer to spread. And so we went back to the work that we knew. And so maybe what we're seeing is maybe this is an RNA storage site, and we wanted to understand how this might work. And that little dark area is maybe where the RNA is assembling. So two amazing scientists in my lab, Dr. Sunjin Park and Dr. Randall Dawn, teamed up together to look at to see if the RNAs were in this midbody structure here. And this actually is assembled in early telophase, but becomes more mature in late telophase. Since then, people also have done genomic sequencing on the MBRs, but I'm going to tell you the story about this midbody in late telophase. And so the way we do this in the lab is we synchronize the cells at each different stages in the cell cycle, and we send them off for sequencing. And we're particularly interested in things in the midbody, but in order to confirm what's in the midbody, we often have to compare that to things in different stages of the cell cycle, so we know it's very specific for the midbody. And the reason why myself and scientists do that is you want to be certain that you know that there's certain proteins that are only at this stage. And so we do this comparative analysis so we know that we're studying things that are unique to the midbody. And one example is this green protein is found in cells that are in interphase, where it's also found in the midbody. So we're not so interested in that green protein, but we're interested in something like this purple protein, which would be something unique in the midbody. So long story short, in collaboration with Mike Blower's lab, who is at Harvard and now is at Boston University, we sequenced the RNAs that were in there, and we ended up with a cluster of RNAs that was quite surprising. And if any of you out there know anything interesting about these blue genes here, FOS is a very famous oncogene that causes cancer. And so we were excited to find these because perhaps these RNAs are actually signals to tell other cells what to do. And so in order to prove that these actually were at the midbody, we actually have to label them. And so we took 4 of these RNAs, and because the imaging of RNA is very expensive, we chose 4 of them to see if they were localized to the midbody in a unique pattern. And we chose these transcription factors, which are in blue because they're known oncogenes. The other one's a known stem cell factor, which is KLF4. And then KIF23, which is this famous protein of the midbody, also ends up at the RNA in the midbody. And so how do these RNAs localize? So we use this technique called a probe called Ahna Skop. It allows us to see the RNAs. I call it a molecular chandelier. It binds to the RNA and lights it up. It's kind of like Christmas lights in the cell. And so this took us several years to do this because this is about a micron in size that we're looking at between the cells. So it's very small. And so we were able to do that. And we use a tag that labels all RNAs, and this is called poly A. So we were able to see RNAs there. And then the cluster of those 4 genes that I told you about, these 4 RNAs, they all localize into the midbody. This was a surprise to people who worked on mitosis because people didn't realize that RNAs are assembling here. And so this was the first hint that there was something more than meets the eye to the cell division process. Is the cell is now assembling RNA at this midbody site and creating this large vesicle, this MBR, that is full of RNA. So this was a totally new and unexpected discovery. The other interesting thing that we found is that if you have RNAs there and RBPs, how is it maintained at that site? So when we looked at our sequencing, I got really excited about a protein called ARC. This protein called ARC has been in national news, but people may not know about it, but it's quite famous, has been in National Geographic as well. And Jason Shepard is quite famous for this, who's at the University of Utah. So I called Jason and I said, we found ARC in the midbody. And of course he said, what the heck is a midbody? Because a lot of people don't really appreciate what a midbody is or that it exists. And he hadn't noticed it was in the midbody. And so ARC protein allows us to have memory. And I was fascinated by this story is because the mice are mutated in ARC actually lose their memory. And what I didn't also appreciate is neurons actually send little vesicles with ARC and RNA in them, and they talk to each other. And when you don't have this protein, you lose your memory. And so perhaps ARC is involved in a cell division memory, and we wanted to look at that. And so we got an antibody from Jason, and sure enough, we found that ARC is in the midbody, in that little dark area where we found the RNA. And what is even more fascinating is that when you knock down ARC here, is that here's the RNA in a control cell, as we label it in green here. When you knock down ARC, cell division is totally fine, but what you end up with is you don't have any RNAs at all in the midbody or in the MBR. And if you knock out another protein involved in the midbody, you still see RNA. So this is very specific to this protein called ARC. And so perhaps this is a mechanism, this protein called ARC is involved in maintaining RNA-based memories that are involved in how cells communicate with each other. And it's not just in neurons. And so we use ARC quite often in lab to understand this process. And so the next question scientists often ask, if you have RNAs, maybe are they translated? And you may not understand what translation is, but I'll give you a little background of what it is and why it's important. So many of you know about DNA, right? So DNA is like the cookbook. It's like all the recipes that build your body, right? But if you want to make a certain protein, you need to release some RNAs out of the nucleus. And this is actually like a photocopy of one of your recipes in your cookbook. And your cell wants, your body needs this photocopy, so it only can make one thing, and it makes a protein that is required, maybe some part of your brain. It makes the actual dish. And so this is the event that's called translation. And so RNA gets translated into protein. And so if we have a lot of RNA in the midbody, we want to know, is it translated and maybe used in other cells? And so to do that, we synchronized the cells and we added a drug that allows us to see translation at the molecular level, at something at the ribosome. This is a drug called pyrimycin, and you add it into the cells, and then you add a little fluorescent tag onto it, and you look to see where is the translation. And so we went to do this, and I didn't know what to expect. I'd been studying mitosis and my whole life, and I never would have expected how clean this would have been. And so this is the day. So Sungjin came right in my office, and this is where the translation event is. This is the zoom up of the cell. So the cells normally translate in their cytoplasm, but there's a distinct site at the midbody that translates. And this was me in my office because I did not believe that. That was too good to be true. And this was a very distinct site. And then Sungjin did a reconstruction of this. And this is actually a ring, what we believe is around the RNA. And so as the RNAs leave, they immediately get translated and they're within this midbody structure. So this was quite exciting for us to find. And so what we noticed, and Sungjin had noticed, is that only in this late telophase stage is all of these are little rings in mitosis. And then when that MBR gets generated, they are also translating. And so, but it's very distinct to this phase in mitosis. And if you remember back to earlier in my talk, the ataxin-2 protein caused a delay. We believe it's acting at this stage as there's a delay in this translation event, perhaps in the ataxin-2 knockout, but this was indicative is that we were seeing this similar kinds of things. As late telophase is extremely important. This is when the translation event happens. What we found is in all cell types, whether you're in your eye or in your HeLa cells, which are coming from cervical cells, in hamster ovary cells, or even in neural progenitor cells, they all actually translate. So this is a very well conserved a big finding that no one really knew, that there are these distinct vesicles that are actually translating, and it doesn't matter what cell type doing, it's coming from these mid-bodies. And they have this translation activity. And so the oddity of this, as people understand, is that cells translate, but now we have this little mini cell, which I often call this lunar module. It's a little mini cell, which also has all of these machinery to synthesize proteins in this. So this is a new concept in science, is that it can synthesize proteins, but it has an RNA genome. And so the cell has the DNA genome, and this midbody now has an RNA genome that cells are shedding and other cells are communicating with it, perhaps. And so here's an image of this. So that's the cell in the division where it's translating. When the cell separates, you see that little vesicle has this translation activity, and other cells are taking this up, and it has this, this little packet of information that other cells take up, and perhaps using the oncogenes that are packaged in it is changing the proliferation state of that cell. The other interesting hint is that we found that there was a timing to this translation, because we found a protein that in early telophase looked like it was here on the bridge, but ended up in a ring. And so we think this domain of translation, where all these ribosomes are sitting, is happening in at late telophase stage. And we found this out is because we noticed is that this is the DNA state. There's no translation, and the this red label is the lamin, which is the nuclear envelope is forming, which this is all forming at the end of mitosis. And that right as you get to late telophase, the translation event lights up as soon as this nuclear envelope and the cells have separated. You get this translation event. And it suggests to us that there's a timing to this whole order of RNA assembly, and that you get an activity of protein right at that moment before the cells separate. And so all these RNAs that we found here, and I'm going to circle the ones in green that we're going to look at, are any of these translated? And what Sunjin found is that all of the 4 of them that we picked to look at, all of them only get turned on at this G1, and then after mitosis, and they're active. And so that suggested to us that the RNAs that are being packaged there are being translated late at the stage. We still don't understand why, the timing of this, and why this is important, and something my lab is looking into. So what have I told you today? Well, I told you 2 things, is that this midbody is actively translating. It is an RNA-based granule we call a midbody granule, an MB granule, and we believe it's important for cell communicate, and it had been lost to science because many people thought this was the garbage can, certainly. And what I'd show you today is that RNA gets assembled here. It then gets translated here in magenta, and then the cell cleaves and produces 3 things. And this 3rd thing can go to an adjacent cell, and we believe it dumps its RNA. So we're doing experiments in the lab to show this and try to figure this out. And so how might it influence other cells? And so we have a couple ideas in the lab about how this may happen. The cell in this first case may bring in an mRNA, and it may be translated, and that cell can change its fate or not. Or it can bring in things called these small microRNAs, which actually can knock down genes. So you can either turn on genes or knock them off. And that's what we're trying to test in the laboratory now. And we think this is because midbodies have been thought to actually do several things. If you take up a midbody, one cell changes its fate, and that's this polarity idea. The other idea is that if you dump a midbody on a naive cell, you can change its proliferation rate. So this is basically the cancer idea. So if we're bringing in RNAs, it's telling the other cell to proliferate either out of control or to not proliferate, for example. Midbodies have also been shown to be important for the building of cilia, which allows you, for example, in your gut to take up nutrients. And there's similar proteins involved in midbodies as in cilia. And also in neuronal differentiation. So midbodies have been shown to be involved in that, and a lot of proteins involved. And midbodies are also found at synapses and how information gets transferred. So we're really excited about trying to figure out what this MBR does, and does it play a role in all these things. And particularly my lab is interested in the cancer side. And my lab has been studying both cervical cancer and breast cancer. and looking at that, does the midbody confer this proliferation thing? So when the paper had come out that I've just told you about, it got picked up as perhaps the midbody. This is the surprise. So it was a garbage can. It was, as you could see here, there was a headline: trash can of the cell responsible for spreading cancer. New study warns. What is it? Well, I've just told you what it is. It is this remnant of division that was lost to science that people thought was a garbage can. And so we're really excited about trying to figure this out and understand, is it transferring information and causing cancer to spread within your bloodstream? And understanding just normally how cells utilize it for information transfer. And RNA is particularly extremely important for sending signals and information transfer. And you probably heard in the news, people think RNA is bad. We would not be alive if it was RNA. And as I've told you, the story today is RNA is absolutely essential for just about everything, and particularly in the case of cells communicating with each other. So if you don't communicate with each other, your cells in your body are not going to work properly. And RNA is a really big part of that. And so understanding this process is very essential to understanding and helping people with disease. And so particularly in my lab, studying the cancer side of this story. And so I'd like to leave you with this story as I started, at the beginning of here, really passionate about the midbody. And I hope I convince you that instead of the garbage can, the midbody is a gold mine of information. And we're trying to understand, and the gold is the RNA here, and understanding how these RNAs function and communicate with each other. I'd like to thank the NIH and the NSF for their support, and all of my lab members here, particularly Sunjin, who did this work. And I have a wonderful group of past and present members that have contributed to this body of work that I have here. And we don't work in a vacuum. I think a lot of people think scientists spend a lot of time by themselves, but we work with a lot of other people, and we have a lot of midbodies around the world that we help and communicate with each other that help to push the science forward. So I'd like to thank all of them and all of their funding that they have to contribute to this project. And with that, I can certainly take questions in the chat, and I appreciate your time. Thank you.
That was such a good chat. Thank you so much. I think we already have, we're ready for our question and answer portion. And I think we actually already have a question in the chat.
Sure.
If I'm looking at it, if you could design one experiment with no limits, what would you like to learn about the midbody? And that was from Ian Chapman.
Yep. Thanks, Ian, for that question. That's a great question. I think one of the big experiments is, can we transfer RNAs to other cells? And then if we knock out the ARC protein, right? So can we make vesicles, if you mutate ARC, that don't have any RNA in them, and then cells will not proliferate? So we're trying to prove that perhaps midbodies actually are this conferent. So that's kind of what we're doing here. What we're doing to do that question, it's very technically challenging, is we color midbody RNAs with one tag and we dump it. And if it dumps in the cell and the cell changes color, we kind of have this working. We're trying to fix this. It's a color change experiment. So if the RNA is dumped and it changed the proliferation rate, we want to see that, and we can see that live on the microscope. So I'm all about seeing is believing. And so I like to see things. And so that's one of my kind of dream experiments that we're actually trying to work out, but it's technically challenging. So again, these are 1 to 2 microns in size. They're quite small. The cells are about 20 microns. So they're quite small, and they're hard to see and hard to catch. And so, but that's one of my dream experiments, is can you track the RNAs that are coming from the midbody going to the other cell? And what is the fate of that cell that takes it up, right? Does it change the fate? Does it become a cancer cell or not? And so that's what I like to do. Great question, Ian.
That is a great question. What color would you choose? If you can make it any color.
You can make it any color, right? So you can make it any color.
All right, so our next question is going to be from Tiara, and it is, when midbodies are added to cancer cells, are they programmed in some way to promote or abolish proliferation? Or is that the canonical function of midbodies are able to simply communicate proliferation?
Yeah. it hasn't been proved yet, but there was a paper that someone dumped midbodies on to naive HeLa cells. They're already cancerous, and they changed their proliferation rate. It was minimal, but we're trying to figure out how to optimize that experiment, and other people are trying this too. And so it's a hint that perhaps this is how cancer spreads. So if you add the RNAs that are coming from the midbody are oncogenic, they can transfer that information and change that cell to become cancerous or not, right? But the cells also can take up midbodies and perhaps not do anything with it, right? So it's kind of, I would say it's almost like text messages, right? So you can either consume the text messages or you can ignore it. And I can think that that's exactly kind of how the midbody works. And the cells, lots of cells in different tissues behave in that way. So some tissues might want to take that information up and do something with it, and other tissues might not take it up, or they might get rid of it and discard it. And so there's two pathways that can happen, but it's a great question. It's just a game of cellular communication, right? And it's done through RNAs.
Perfect. I think along those lines, we have another question, which is a two-parter from Abhishek, and they were wondering how midbody retention is conferring the cancerous phenotype, but also which cell after division gets the midbody and what can determine that.
That's a great question. So we don't understand. It was noted many years ago that perhaps cancerous cells they actually have a veracity to eat midbody remnants. And so they take up a lot. So people would see tons of midbody remnants in cancerous cells, and then cells that were like stem cells didn't care to eat them. So stem cells don't like these midbodies. So maybe that's why they're involved in oh my gosh, I'm having a brain fart. So stem cells don't like to eat them, but cancer cells eat a lot of them. And so perhaps this is why they're cancerous, is because they have a lot of this information. They don't know what to do with it, and it's unregulated, right? So, and the next question was, which cells after takes it? So we don't understand that at all. And that is a great question. We think there's surface markers on the midbody that it's a cell. The same type of cell has to communicate, like a skin cell will only talk to a skin cell, and there's a surface marker that tells it which is which. And so we're trying to understand that by doing proteomic experiments to actually figure out what's on the surface of the tissue and the cell and what's on the MBR. And so we believe that's the case. There's some hints that that might be the case, but it hasn't been studied in different cell types. That's a great question.
I think these questions are following a really good flow, because the next one is there a way for the body to detect if the MBR is bad? And can MBRs be a way to introduce genetic fixes into the cells?
Yes. And so to figure out if a midbody is bad, we have to take a normal tissue and sequence the midbody remnants and go, okay, in a normal cell, supposedly, right? I mean, we also don't know what is normal, right? So a normal cell, if you take a healthy cell, what is on the surface of a midbody remnant, right? And if you take a diseased midbody remnant that's coming from a certain cancer that, say, has a certain marker, there is several things in the literature that tell that are on vesicles that if you have a mutation, you can surveil it, and this gets put on the surface of them, right? So we don't know the answer to different types of cancers, but these are things that we're kind of doing in my lab now, is sequencing different types of cancers, and do the cells have different surface markers, and do the midbody remnants do? So once we know that, then we could figure this out. But that's another great question.
We're just going to keep it rolling with the great questions.
Sure.
So someone was curious about remnant clearing mechanisms. And are many of these age-related diseases, or are these mechanisms dysregulated in diseases?
Yeah. So the clearing mechanisms for the remnant is the remnants will be endocytosed, and they get taken up, and they're often degraded in the lysosome. And the lysosomal compartment is where people see them. And so if a cell takes them up, they would degrade them like they would degrade some foreign material. And so I think that's the mechanism for theirs. And then the next question was about as many of these are age. Yeah. So lots of diseases are associated with clearing of things in the lysosome. So there's things called lysosomal disorders that have shown up in human diseases, but perhaps the midbody's in part of this, and we don't understand it entirely. But maybe in cancerous case, you build up a lot of midbody remnants and you don't know how to regulate them. The cell doesn't do it, and that's how you got to kind of get cancer. Yeah, I mean, lots of. I was just reading a paper today about, so the Alzheimer's case is inflammation is a major cause of cell division defects in the brain, which is quite interesting, which causes leads up to a lot of these Alzheimer's patients. And so perhaps the midbody is involved in, if cells are dividing and there's cell division defects that are needed in the brain, there's dysregulation there.
And now people are wondering, is it tissue only, or is there possible research for blood cancers in relation to midbodies?
Yeah, so wherever you have a cell division, right? So it's not just tissue. So anytime you have a cell division, you'll produce a midbody remnant. And so these vesicles are being generated, taken up, and regulated. We don't understand how all this works, but it's possible. And I don't know exactly. We've been working and looking at platelets lately, and platelets don't divide normally. And we don't know if they actually produce midbodies. They produce little vesicles of RNA of them, but we don't understand much about blood cancer. There's very little cell biology on the blood. It's actually quite remarkable. As much as we look at it for surveillance, there's very little known about what's happening in the blood and all the different types of things that are in blood that divide. And so it's possible that that's also the case, but that's a good question too.
Thank you. I didn't ask it, but thank you whoever asked it. And I think this next question is from Gracie. Are the RNAs being synthesized in the MBR, or are they being trafficked there?
So we think they're trafficked there, and the kinesin protein and this ARC protein, if you knock them out, the RNAs don't get there. And the kinesin protein is a motor protein that moves. We think it moving the RNA there, and so that's what we think. It's actually the whole process of mitosis. Instead of just building two cells, is actually to assemble this massive amount of RNA in the midbody, right? So knowing that this exists now, we have to get lots of researchers to try to figure out how all this works, right? So, and getting there. But we definitely see RNAs early in anaphase and then gets pushed into the midbody. So we think there's an active mechanism to traffic it there. How exactly all it works, we don't totally understand. But those RNA binding proteins that we found, those bind RNA and bring in there. So how does 100 RNA binding proteins, they're bringing all different types of RNAs to the midbody. So that process of mitosis is doing a lot of things, but in addition, pulling RNA there and building this giant vesicle full of RNA. But yeah, all of these questions are amazing. You should all be in my laboratory. This is, if you can understand this from this talk, right? These are the kind of things we're asking the question, and you could be a scientist, right? So asking these great questions is these are almost hitting all of my grants I'm applying for. So let's hope.
Perfect.
So I really appreciate all the input. Is there a last?
And so now there is another question, and then I think we have a final question after this. So do all cells package the same RNAs, or is this tissue specific?
It's definitely what we find. We have a paper coming out soon is tissue specific. There's tissue specific midbody remnants. And so that is another surprise to me, is that there is a core set of genes that are common between all midbody remnants. And then if you're in a certain tissue, whether it's cervical tissue or breast tissue, you are packaging RNAs differently into this structure. And so if you're in a certain tissue, you want to be kind of the same. And so that's how we think this happenings, right? But that's also important because we can surveil that and predict where that midbody came from based off of that information.
Thank you. And I actually have our last quick question, because I've always been fascinated on how you branch art and science together. And I was wondering if you being an artist has influenced your science progression in any way, and if so, how?
Well, I think I started grad school right as PowerPoint was invented. So I used to give talks off old slides, right? So the old-fashioned slide decks. I think for me, is I was like a kid in the candy store because I was able to use my artistic sense to put together, a communication of science. And I think art has really helped me. My art background has really helped me understand how do I communicate science more effectively. Also, there's a lot of creativity that goes into slide decks. You don't have to be boring at all. And you can see that using color is also really helpful. What's helpful for teaching people and connecting ideas. You can see I use those little tips. This is all coming from art, right? So art is essential for how we communicate science, right? I use art in other ways to make science more accessible with other projects I work on in my lab and different things. But I think for me, it's absolutely essential for science communication. It's also helped me understand even just microscopy. The colors that I see make me understand things better when in the microscope. So having a art background is extremely helpful in discovery and just observation, right? In that part. And again, the drawings that you see in my slides, they're often drawings we draw, right? So we draw them and then we do them in Illustrator or work with artists to draw those things. And so art is absolutely essential in communicating our ideas to other scientists, but also to the public and making them understand things more clearly.
Thank you so much for that answer. And I just want to thank you again so much, Dr. Skop, for being such an inspiring and an insightful talk today and being able to share your story with us. We're incredibly grateful for the time, knowledge, and the passion that you were able to share with us tonight. And I want to thank everyone who joined for this milestone event, our very first. Your participation and curiosity means a lot to us and our Atlanta team. And as a quick reminder, we are a national nonprofit dedicated to making science accessible and engaging. So we're excited to bring more events like this in the future, but hopefully in person. And if you enjoyed tonight's event, we'd love for you to stay connected on our socials and our upcoming events.
Yep. Thank you all for coming. Appreciate it.
Oh,